All right, Ninja Nerds, so what we're going to do in this video is we're going to talk about the brain-testicular axis. Okay, so first off, let's go ahead and look at what specific organs are involved here. So if you look here, what I have zoomed in on right here, this is the hypothalamus. So the hypothalamus is basically the master endocrine regulator, right? Because he controls your pituitary gland. How does it do that? So if we look here at the hypothalamus, so again, what is this right here? This is the hypothalamus. Just has it consists of a hypothalamus consists of a bunch of gray matter. So basically these nuclei that are lodged within what's called the diencephalon of the cerebrum, right? So it's a component of the diencephalon. The hypothalamus has these specific sets of nuclei that are responsible for secreting a hormone that's going to regulate a lot of this cycle here. What is this hormone here called? Well, the hormone that these two nuclei are actually secreting here is called gonadotropin releasing hormone, GnRH. Okay, so that's GnRH. And what nuclei are secreting this? This is actually going to be, let's say that this one over here is the preoptic nucleus. Okay, so the preoptic nucleus is secreting it. And let's say that this green nuclei over here that's secreting this GnRH is actually called the arcuate nucleus. So these are two nuclei that are very, very important in being able to secrete gonadotropin releasing hormone. Now, what does that gonadotropin releasing hormone do? You know, it actually moves through this nice little portal system down through here. It's called the hypophyseal portal system. It's a blood vessel connection. And look what it's doing. It circulates down through that, and it binds onto these receptors on these specific cells. What are these cells here called? These cells are called gonadotropes. So they're called gonadotropes, okay, or troughs. But what this gonadotropin releasing hormone does is it binds onto the receptors on these gonadotropes and triggers the production of two very important hormones. One of the hormones is called luteinizing hormone, and the other hormone that's secreted is called follicle stimulating hormone. Okay? So one's luteinizing hormone and the other one's follicle stimulating hormone. Now, before we talk about what these guys do, we have to talk about the interaction that's actually all the physiological mechanisms that are occurring in this area. What is this structure that I'm drawing right now? You know, in the testes, you have this structure called the seminiferous tubules. The seminiferous tubules are very, very important. Why? Because that's where spermatogenesis is occurring. What is spermatogenesis? It's sperm production, okay? So what I'm doing is I'm zooming in on one seminiferous tubule. Now, seminiferous tubule is primarily made up of these very, very important cells. You see these green cells right here? These green cells are very important cells, and we're going to call these green cells, we're going to call these Sertoli cells, or I can call them sustenacular cells, and sometimes they even call them just nurse cells. But either way, you can pick which one you like the best. Either way, one of these three cells is the name of these cells that are making up the seminiferous tubule. Now, really important, in between them, they have these proteins that are linking them together and keeping them very, very tight to one another or adhering one another, right? What are these things called? These are called tight junctions or adherence junctions. They're actually two different types. So we have, what is it again? Tight junctions. And we have other ones which are called adherence junctions. Why am I mentioning this? Okay, and we'd also have these ones right here, right? So this would be a junction here, and this would be one joining right here. These are very, very important proteins. They're actually called claudins and occludin proteins, and they're very, very important at being able to link these actual cells together. If it's tight junctions, it's claudins and occludins. If it's adherence, it's actually cadherin molecules. But neither here nor there, these molecules are linking the cells tightly to one another. What is that doing? That's separating this actual, if you will, seminiferous tubule into two compartments. One compartment over here we're going to call the basal compartment because it's closest. You see these black cells right here consisting of connective tissue cells? These black cells here is the basal lamina. And then right here is the blood vessels that are going to be supplying the seminiferous tubules. So the basal lamina, look, if it's on this side, it's the basal compartment. If it's on this side, it's closer to this central space, which is called the lumen. So this is called the adluminal compartment. Okay, specifically this part right here. So basal compartment, and then adluminal compartment. And then down here is the lumen, okay? Now let's see what's actually happening. In this area right here, we have some cells that are actually going to be formed here. These are stem cells, very, very important stem cells. Okay, let's look at these guys right here. So let's say this one is a diploid stem cell, and it's 2N. So that's what diploid means, right? It has 46 chromosomes, 23 maternal, 23 paternal. What happens is this spermatogonium, right? So what am I calling this? It's a spermatogonium. Heck of a name, right? And then what does it do? It splits into 2 different types of cells. But look what these cells are here. Look, they're 2N and 2N. Whenever I start with a stem cell and I separate it into 2 identical daughter cells, that's called mitosis. So what just happened right here? This event right here is called mitosis. Okay? Now, we're actually going to rename these now. We're going to rename this one a type A cell. So we're going to call this one a type A cell. And we're going to call this one a type B cell. What happens is the type A cell that's formed by mitosis from this spermatogonium is actually going to come back up. So it's actually going to, look at this, it comes back up to the basal lamina and it just actually gets reused. It becomes the next spermatogonium and just continues to keep replicating. The B actually is going to move out of this basal compartment into the adluminal compartment. How? There's a lot of chemicals that are secreted, right? These actual cells, these spermatogonium cells and other different cells nearby are secreting chemicals. And what these chemicals are doing is they're telling the sustentacular cells to open up the tight junctions, kind of like a gate. Why do we need these tight junctions there anyway? What's their significance? It's not just to hold the cells tightly together. You want to know what's really important about it? If we separate these compartments, when I'm making sperm, because that's what I'm doing, I'm making sperm down here. That sperm is developed when? When are we actually making sperm? We're making it during the puberty process, right? Now, our immune system is actually being able to, it has to recognize any type of self-antigens in the actual thymus gland, right? Our T cells. Our T cells undergo that selective process, right? But it occurs before puberty. So whenever someone starts making sperm, it's at puberty. If by chance, let's say that they have a sperm cell. Let's say here's a sperm cell. And some of the antigens from the sperm cell leak out into the blood. What is it going to do? It's going to activate the immune system, and the immune system is going to start coming and attacking our own testes, right? That's not good. So what these tight junctions between these Sertoli cells are doing is it's forming a barrier between the blood and the testes. So we call that the blood-testis barrier. Okay? And the reason why is you want to prevent these sperm cells or their antigens from leaking into the bloodstream and activating our immune system. Okay. Now, these chemicals that are secreted, what happens? It opens up these tight junctions. So now the tight junctions open up. And now look what happens here. So now these tight junctions are open. This type B cell is going to move into the adluminal compartment now. So now it moves into the adluminal compartment. Once it moves into the adluminal compartment, it's no longer considered to be a type B cell. We actually are going to call this one a primary spermatocyte. So it's called a primary spermatocyte. Okay? Let's redraw him here. And again, what state is this guy in? He's 2N, and what was this one? 2N. Here's where it's going to get interesting. So we're just going to put here primary spermatocyte, but I'm going to put 2N here. He's going to undergo a process where he's going to split into 2 different cells. But look what these cells are now. Holy frick, they're haploid. So now this was diploid, and I went to haploid. Whenever you go from a stem cell that's diploid to 2 daughter cells that are haploid, this is called meiosis 1. Meiosis 1. Okay? But this was a primary spermatocyte. He divided into 2 of these cells. What should these be called? Secondary spermatocytes. So these are called secondary spermatocytes. Okay. We're on a roll, guys. Hang in there with me, okay? Now what happens? These secondary spermatocytes, look what they do. They're like, hey, we're not done. We're going to keep frickin' dividing. And look what he divides into. He divides into a haploid. He divides into a haploid. he divides into a haploid, and he divides into a haploid. They're fricking things up here, right? Now look, we got 4 haploid daughter cells. Whenever you go from 2 haploid stem cells to 4 haploid daughter cells, that is called meiosis II. Meiosis II. Okay? What's actually happening is that this primary spermatocyte, it's replicating. So for example here, if I were to show you like really kind of like a little bit real quick review of what's happening here. Let's say here's the chromosome. What's happening is you're replicating, so originally you only had 1 chromosome there. You replicate it, right? Still have 1 chromosome, but it's just a replicated version. What happens is, let me actually make this cell bigger here, is I could take a chromosome, let's say this is chromosome number 4 from the maternal side. And over here I draw the paternal chromosome of 4. What happens is these are called dyads, right? They come together and fuse like right around the actual tips. And they do what's called a crossover or synapsis, where they cross over some of their genetic material. Now in mitosis, what you do is you separate them at the centromere. But in meiosis, I'm not separating them at the centromere. So when these guys link together, they're actually going to link together and do what's called recombination or crossover or synapsis. When they're linked, they're called a tetrad. But individually, they're called dyads. Why am I mentioning that? So that you can really understand what's really happening versus mitosis. So in mitosis, you separate these at the centromere. So you get 2 chromosomes going to these cells and then 1 going to that cell too, right? If it was mitosis. In meiosis, this whole dyad is going to 1 cell. This whole dyad is going into the other cell. The only thing that's happening that's different is that some of the genetic material is crossing over. So for example here, just to finish this off, let's say I had 1 cell over here, 1 cell over here. All that's happening is I'm going to redraw this chromosome over here. Look what's different. I'm going to draw a piece of blue in here. That piece of blue was from that chromosome. But now I'm going to redraw this blue chromosome over here. You see how we got 2 dyads in each corner? All that happened is I swapped a little bit of the genetic material right there. That's all that's happening. And then these guys will get separated. So now we're going into 4 haploid. Guess what? I'm going to separate that at the centromere and that at the centromere. I'm going to separate that at the centromere, and then I'm going to get how many of these chromosomes? I'm going to get 1 here, 1 here, 1 here, 1 here. That's how I get these 4 haploid daughter cells. These are now called spermatids. So these are called spermatids. Then what happens is these spermatids are going to turn into some more motile and functional sperm cells. So now look what's going to happen here. You see how now they're becoming more well-formed sperm? They have a head, they have a mid-piece, they have a tail, they have the acrosome, all that stuff. So when they're going from spermatids to, what are these guys called? These are called spermatozoa. Spermatozoa. When you go from spermatids to spermatozoa, this is a specific process. They call it spermiogenesis. So it's called spermiogenesis. Now, this whole process, starting from a primary spermatocyte to a secondary spermatocyte to spermatids to spermatozoa, that whole process in general is called spermatogenesis though. So if I were to highlight from here, all the way from this point here, all the way down, this is called spermatogenesis. Spermatogenesis. Okay? Technically what you could do is you could separate right here, you see where it comes to spermatids? I could make a little line there, and I could say from primary to the actually getting to the point of like the spermatids is called early spermatogenesis, and then from the spermatids to the spermatozoa, I could call it late spermatogenesis. But I'm just going to say this whole thing is spermatogenesis, and the spermatids to the spermatozoa is spermiogenesis. Okay? Now that we've done that, I have to explain a couple more things about these sustenacular cells. So you notice how all this stuff is happening. It's not happening on its own. There's got to be these FSH and this LH. They got to be doing something. Let's see what they're doing now. Okay. So over here, let's focus on LH first. Let's see what LH is doing first, because he's going to be doing some really, really important stuff with a very, very important metabolic hormone for males. So luteinizing hormone comes over here and it acts on a specific cell. What is this cell here called? This specific cell is actually called a Leydig cell or an interstitial cell. So this cell right here is called the Leydig cells, or again, the interstitial cells. What luteinizing hormone does is it acts on this Leydig cell, which triggers some specific signals to the nucleus. And it makes a specific it makes these actual enzymes, right? So it's actually going to make some specific enzymes. What is he going to do? This enzyme is going to catalyze a specific reaction. He's going to have his hand out here, and his hand is actually going to catalyze the conversion of cholesterol all the way to testosterone. To go through the whole pathway is not necessary. But basically what this enzyme is doing is, these enzymes, it's helping to catalyze the conversion of cholesterol to testosterone. And then where's the testosterone going to go? Where's it going to go? It's going to come out here, and it's going to act in this vicinity. And I'm going to explain how in a second. Okay, but before we do that, we have to say what FSH is doing over here. So let's draw a receptor over here for FSH. Let's say here is this receptor for FSH. And look what happens here. The FSH is going to come over here, and it's going to do what? It's going to bind into this receptor. Similar thing. It's going to send signals to the nucleus, and this nucleus is actually going to synthesize a new protein. And this new protein that's actually going to be made is called androgen bonding protein. And this androgen bonding protein is actually going to get bound, it's actually going to get secreted out here. Now I'm going to explain what happens. So this whole process for spermatogenesis, it requires the presence of two different molecules. This one right here, which is called testosterone, and this molecule here, which is called androgen bonding protein. Now, what's the function of androgen bonding protein? Well, you know testosterone is actually very lipophilic, which means it loves lipids. So it's not very good at dissolving into a water-soluble-like environment. Androgen bonding protein helps testosterone to be less lipophilic. So it actually is going to want to stay in this vicinity. And if testosterone is very concentrated in this vicinity, it's going to help for these sperm cells to develop. So it's going to help the spermatogenic process. So testosterone is very important in all of these steps here. It drives this step, and this step, and this step, right? So it's stimulating all of these processes of spermatogenesis. But all androgen bonding protein does is it helps to be able to keep the testosterone in this vicinity and keep it very, very soluble and highly concentrated. Now, sustenacular cells, or the Sertoli cells, they're not just opening up their tight junctions, they're also doing some other stuff in here. They're passing a lot of nutrients because these cells are going to go on a very long journey to the epididymis. So they have to have enough nutrients and metabolic products to be able to carry out that process. So these sustenacular cells are providing nutrients to the sperm cells. You know, you see this process where they went from spermatids to spermatozoa? It also helps this spermiogenic process. Because what's happening is, it's taking this sperm, which was just like a big immotile blob, and look what it turns it into. It turns it into a well-defined sperm cell that has very, very important structures that I need to talk about here because it's very, very essential. All right, this part right here is called the head of the sperm. This part right here is called the midpiece. And then this last part back here is called the tail. And each one of these consists of some very specific structures. The head obviously consists of the DNA, but there's another thing in there which is extremely important. Without this, we would not be able to form fertilized ovums, right? This green structure I drew is what's called an acrosome, and it's very, very essential for it has hydrolytic enzymes that'll eat through specific types of things within the egg. The midpiece consists of mitochondria. Okay, so let me actually write this down. What was that green structure? It was called the acrosome. Then this midpiece consists of mitochondria. And the mitochondria is extremely important because that's what's going to have it's going to utilize the ATP. It's actually going to form ATP from different types of maybe carbohydrate molecules or different types of nutrient sources that the Sertoli cells are providing. And then the tail is actually going to be considered to be what? You actually call this flagella, right? So it's going to be made up of microtubules, and then at the base of it, you'll have what's called a basal body, right? And this is in this flagella is in a 9+2 arrangement. Okay, so this right here is spermatogenesis, going from an immotile spermatid to a little bit, not yet, but a little bit motile sperm cell. These Sertoli cells help this process to occur, that spermogenic process. What else? Okay, you know testosterone doesn't just go right to the seminiferous tubules to help sperm production. It also goes into the blood and acts on different tissues also. So it can also act on other tissues that we're not going to discuss. But I'll just put here, other tissues. Okay? And it can exert its effects on that. But, like anything, elevated testosterone levels are obviously not good for the body. So let's say that there's high T levels, testosterone. What's going to happen? Testosterone is actually going to come up here. It's going to go up to the actual hypothalamus. Watch this. And it's going to exert a negative feedback mechanism on the hypothalamus. You know where else it can exert a negative feedback? Here at the anterior pituitary. What's that going to do? If you inhibit this hypothalamus, you're going to make less GnRH. If you make less GnRH, you're going to make less luteinizing hormone. Less luteinizing hormone makes less testosterone. Okay? That's important. Now, who controls the FSH? You see these little black little sensors here? They're picking up sperm levels. So these sustenacular cells they're weird. They have the ability to pick up levels of sperm. If the sperm starts getting really, really high, the high sperm production, these sustenacular cells will be able to detect that. When they detect that, they actually secrete a specific chemical. So let's say this actual Sertoli cell picks up that signal. Right? So it picks up that signal, sends signals to the nucleus, and the nucleus makes a hormone that's called inhibin. And what does inhibin do? It comes up here, watch this now. It's going to come up and it's going to exert a negative feedback mechanism on the hypothalamus, and it's going to exert a negative feedback mechanism on the anterior pituitary. What's that going to do? That's going to inhibit the release of GnRH. That's going to inhibit the release of FSH. If there's less FSH, you'll have less ABP. If there's less ABP, testosterone won't be concentrated enough, and spermatogenesis will actually decrease. Okay? All right, guys. In this video, we covered a lot of information. I hope it all made sense. Until next time, Ninja Nerds.